Sir, please disable some options on the display.
Now, sir.
Dear participants, respected resource persons, and my dear colleagues, and different colleges of principals, good morning to one and all. Welcome to fourth day of Faculty Development Program sponsored by Acharya Nagarjuna University, Rusa 2.0, on the title Next Generation Drug Discovery Leveraging AI Tools for Advanced Pharmaceutical Research and Regulatory Innovations. Today, we have two sessions. Morning session, the resource person name is Dr. Mohan Gandhi Bonthu Garu, and he's working in JSS College of Pharmacy, Ooty. Welcome you, sir. Next, the second session is going to delivered by Dr. P. Rajeshwari, KLU, Andhra Pradesh. So, one of my colleague, Srimati V. Lakshmi Monica ma'am, will introduce the sir. Thank you, ma'am. Welcome you, ma'am.
Here I am delighted to introduce Dr. Mohan Gandhi Bonthu. Sir is a highly motivated associate professor with extensive experience in teaching and research in pharmacy college. He possesses strong expertise in analytical instrumentation and method development, analytical and bioanalytical. Sir currently working as associate professor in JSS College of Pharmacy, Ooty, Tamil Nadu, and he previously worked as professor in School of Pharmaceutical Sciences, Sandip University, Nashik, Maharashtra, and he worked as a professor in VV Institute of Pharmaceutical Sciences, Gudlavaleru, AP, and associate professor in Sri Vasavi Institute of Pharmaceutical Sciences, and assistant professor in KGRL College of Pharmacy, Bhimavaram, AP, and sir has worked in project training in Glenmark Generics Limited. And sir achievements are, he received Best Presentation Award in Indian Pharmacy Congress 2015 and also received Dr. P.D. Sethi Award for Best Research Article in 2017. Thank you, sir. Thank you.
Thank you, ma'am.
Please
Thank you,
start your session,
for,
sir.
for your kind introduction. Thank you so much. Please confirm whether I'm audible or not. Am I audible, ma'am? Please confirm.
Yes, sir, you are audible.
My screen is visible, right?
Yes.
And my camera is on. Yeah. Thank you so much. Thank you, Monica ma'am, for the kind introduction. First of all, very good morning to everyone. I would personally like to thank Professor and Principal Tejamurti Garu, University College of Pharmaceutical Sciences, Acharya Nagarjuna University, and the organizing committee of 1 week faculty development program sponsored by ANU Rusa 2.1 or 2.0 for inviting me to share my views on this particular platform. And I've been following the lectures or the presentations that are being given by renowned personalities in this field. And all the presentations were very informative, and they are mainly concentrating on AI as per the theme of the FDP. In line with the theme of this particular FDP, I would like to share my presentation on the topic transformative impact of artificial intelligence in pharmaceutical research, advanced applications in pharmaceutical analysis. As introduced, I'm Dr. Mohan Gandhi, Associate Professor, presently working in the Department of Pharmaceutical Analysis, JSS College of Pharmacy, Ooty. As I belong to pharmaceutical analysis background, my presentation would be in line with pharmaceutical analysis, its application, and how we can integrate the AI into pharmaceutical analysis. And for the coming 1 hour or 1 hour 15 minutes, I'll be speaking on these contents. Initially, I'll be touching a bit on the drug discovery, how the journey of drug molecule from laboratory to market, how it travels, and during its journey, what are the different challenges we or the scientists or the analyst face as far as pharmaceutical analysis perspective is concerned, that I'm going to touch. And then I'll be moving into the fundamentals of artificial intelligence. I'll be, taking some 5 or 10 minutes in discussing the fundamentals of artificial intelligence. Then what are the artificial intelligence techniques that are available exclusively for pharmaceutical analysis? That I'm going to discuss in detail, which will be consisting of the analytical method development, spectral interpretation, and how we can integrate AI in bioanalysis, and the hot topic, that is herbal drug standardization. Can we take the help of artificial intelligence in standardizing herbal drugs? I am also going to touch that part even. Then finally, I'll be winding up my presentation with the challenges as well as future aspects or future perspectives. So these are the contents what I'm going to present in my presentations. Now, this slide is speaking about the journey of the drug or the molecule from laboratory to market. This topic or this theme we have been listening to, we have been studying in detail since our B Pharmacy. So this is not a newer topic. Everyone knows about that. But I would like to emphasize few points here. I'm not going to discuss the detail about the drug journey, but I'll be pointing out few things and then try to connect the same thing with the pharmaceutical analysis. The journey, what I'm speaking, is quite fascinating, quite interesting, and quite inspiring. So, which we invest a lot of time, a lot of money, and a lot of manpower. So, for one molecule to be successful, there will be n number of people struggling and working together for this success. There are n number of steps which are involved here. I have taken only few steps. The first one is target identification and screening. Now, you want to target a particular disease, you want to target a particular receptor or the ligand, whatever it may be. That is the initiation what we are trying to do. So once the target is identified, and we try to screen different molecules that are available with you, whether the molecule is fitting with the target or not. This is the initial stage. Once it is done, we will be optimizing the structure of the molecule as per the requirement. We usually call it as SAR, structural activity relationship, or QSAR, where we try to play with the chemical structure of the moiety, thereby can it show a better efficacy or better activity. This would be your second stage. Once it is done, or once a couple of other steps are done, we try to convert the molecule into a formulation by including some excipients or some adjuvants. Then we'll be finally coming up with a formulation. Once the formulation is done, we'll be trying to use or trying to test the formulation on animals first. We usually call it as a preclinical studies. And if the results are promising, we go testing on humans. So we call it as clinical. And then if you are fortunate enough, you may end up with successful molecule, and the same will be released into the market after approval. So this is the journey which involves n number of steps. The point what I'm trying to highlight here is whatever step it might be, we have the steps from A to Z. The moment you complete a step A, we try to understand whether we are moving in the right direction or not. For that, what we do, the moment you complete step A, we try to test, are we getting the result? Are we getting the outcome what we are expecting? This is what we try to do, right? So this is a routine practice from test A to Z. Even after your successful journey, not only for the newer drug formulations, even for the generic drug formulations, you need to analyze the compound. You need to test the compound. So testing, the moment I use the term testing, analysis comes into role. But we will be using the analysis term in a different perspective, basing upon the different steps what we take into consideration. Now, what I'm trying to emphasize here is pharmaceutical analysis is not a separate branch. It is an integral part of your pharma in general and pharma research or drug research in particular. So without pharmaceutical analysis, we don't have the confidence that we are getting the required output or whether we are moving in the right direction or not. So this is where the pharmaceutical analysis plays very, very important role in any pharma related issues, most prominently the drug research. Now, this is what I'm speaking about. So the importance of analysis, be it drug discovery or be it clinical trials or drug approval, whatever it may be, in each and every stage, we need to perform analysis. We need to come up with the results. We need to formulate the report. I'm quoting a few examples here. First one is impurity. To know impurity or to separate the impurity, we'll be using some instruments like HPLC, UPLC, LCMS, or GCMS exclusively to identify the impurities that are present in the given formulation or given API. And then we try to quantify the impurities. Only thing why we are using all these things is without doing all these things, you are not complying with the standards that were given by ICH. This is the one side of the coin. The other part is you are manufacturing one formulation, say a tablet or a capsule, whatever it may be. You are not only testing it in the laboratory, but you are testing it in real world. What is the meaning of real world? Tablet is taken by an individual, say humans. What is happening to the particular tablet? How it is entering into your body? You need to identify the drug concentration in the systemic circulation, say it in the blood. For that purpose, what we need to do? We need to collect the blood, we need to separate the plasma or serum, and then try to find the concentration of the drug, for which we will be using LCMS, which is a gold standard for bioanalysis. Then the formulation should have a certain expiry date. So for which we need to have a shelf life. And when we force it, the formulation or the API would undergo degradation. This is what we call it as a forced degradation studies. So accordingly, we can guess or we can predict what type of packaging materials we need to make, what type of conditions we have to maintain to protect the formulations or API. Okay, so to know that or to analyze that, we need to have a separate method. We call them as SIAMs, which is nothing but stability indicating analytical methods. This is the third vertical. Now, coming to the fourth vertical, obviously, whenever we are formulating or creating some sort of formulation, we need to study how it is getting absorbed into your body, how it is getting metabolized, how it is getting excreted. So all these things has to be taken into consideration. We need its half-life, we need at what rate it is getting eluted out of the body, and accordingly we can optimize the dose. So all these things will be coming under your PK studies. What might be your interest? So I've discussed only 4 parameters or 4 verticals here. We have n number of verticals. Whatever interest it might be, but you should be ending up with an analytical instrument. So we have to perform analysis. So this is the importance of analytical chemistry or pharmaceutical analysis as well as pharma is concerned or drug research is concerned. That is fine as far as analysis is concerned. We agree that analysis is so important, but what is the problem here? We do face a lot of analytical challenges, especially in today's world. What might be the analytical challenge? The first analytical challenge, what I'm trying to quote here is, we are coming up with a complex data sets. What is the meaning of complex data sets? Let me give you a simple example. In good olden days, say some 30 years back or 35 years back, if I want to know how much amount of glucose is present in the blood of human, if he is a diabetic patient, The simple test what you used to conduct is the urine test. We used to collect the urine, and to which we used to add the Bendix reagent. The moment I add Bendix reagent to the urine and try to heat it or try to boil it, it would be showing some sort of color. According to the generation of the color as well as its intensity, I used to predict, okay, this patient is suffering with this level of diabetes. He is having this level of glucose in his blood. This is what we used to follow some 30 years back or 33 years back. Thanks to technology, thanks to the advancement now, with a simple drop of blood, you can know how much amount of glucose is present in your blood. So that is the advancement. But that is positive sides. So now we have traveled from chemical analysis to the instrumental analysis. Previously, we used only chemicals. Now we are using high-end equipment like LCMS, LCMSMS, GCMS, XRD, etc., etc. So that we are coming up with a better, results, better resolution, greater accuracy, greater precision. These are the advantages parts are the, positive things. But we do have a negative things here. What are the negative things we have? The LCMS or HRMS or whatever the latest equipment what we are using, they are giving lot of data, so many parameters, so many features. So you are dumped with the data. You are dumped with the data. But the data what is generated is not ready-made. There should be a person to interpret those results and has to convert the results into easily understandable report. So we need a human interpret all these things. So previously the human used to work on 4 parameters. Now the same human is asked or burdened to carry or to perform interpretation of more than 15 parameters. So don't you think that the analyst is being burdened? Of course, the analyst is being burdened with a lot of complex tasks. This is the first analytical challenge what we are having. The second analytical challenge is multiple component mixture. Now, till now, I'm speaking about a single drug, say Dolo 650. The moment I speak about Dolo 650, it consists of only paracetamol, right? So my main motto is to separate paracetamol from the excipients and from the biological matrix. But in today's treatment, we are not sticking with a single component, but we are dealing with the multiple therapy. The same patient is taking multiple drugs because of different issues. So dealing with different components, separating different components from the target analyte interested is really a Herculean task. This is the second challenge what we are facing right now. The third challenge is about the impurities. Say you are manufacturing or you are synthesizing some APIs. The moment you are synthesizing APIs, you declare that my API is 99% pure. It's absolutely marvelous. Creating 99% API purity is really good, a good part. But are the regulatory authorities satisfied with your, report? No, they are equally interested with the 1%. What you say that 99% is your drug, what about the remaining 1%? So you should give equal importance to the remaining 1% also. What is that remaining 1%? Does it contain impurity? Does it contain, any other intermediate substances? If yes, how many? If you are giving the numbers, you have to give their concentration also. The moment the concentration of the impurity or degradant or intermediate is exceeding 0.1%, you need to characterize it. That is, you have to give each and every information about the structure, the molecular weight, each and everything, along with its toxicity. See, till last generation, we are concerned only with the APIs. But now the regulatory authorities are more interested towards the impurities because impurities are causing more harm than your positive side of your APIs. So this is also one more challenge what right now analysts are facing with. The fourth one, as I mentioned earlier, To develop a method for separation of single component, it's fine. For separation of two components, bit tricky. Imagine developing a method for separating four components, five components in presence of interfering substances, in presence of excipients, in presence of metabolites, and in presence of matrix. Imagine how difficult it would be. So definitely it would be taking a lot of time to optimize the method. This is one more challenge right now I'm speaking about. And finally, interpreting the results. Of course, the instrument is giving the spectrum. Of course, the instrument is giving the chromatogram. And the analyst is the person who is interpreting the results, who is doing the integration. That is really a Herculean task. Let me give you a simple example. If you are working on metabolomics or proteomics, one study using UPLC QTOF instrument would generate thousands of datasets. Datasets consist of thousands and thousands of features. So it will be simply providing you an Excel. So Excel will be consisting of table consisting of 1,000 data, 1,000 values. Who is going to take care of all these things? So definitely it would be a burden to the analyst who is performing. So what I mean to say here is, if there is a sprint behind you, there is a person, learned person behind you who can help you in doing all these extra works, then definitely we can have a better analytical world. So who would be the particular friend? Definitely your AI would be the particular friend. So AI, artificial intelligence, helps to analyze data sets rapidly. It might not be possible for me to integrate or to interpret spectra within 1 minute or within 2 minutes. But the same spectra can be interpreted within fraction of seconds by AI. That is the credibility of AI. The second one is, the moment you feed it with lot many data sets, it try to recognize the pattern. It try to recognize the pattern. So whether it is related to the diseased person, whether it is related to the healthy person, whether you are getting a polar peaks, or whether you are getting a medium polar peaks, or whether you are getting a non-polar peaks, it is able to predict or recognize the patterns. That is what the AI is going to do. The third one is, as you are making or as you're teaching the AI with different types of molecules, the AI can predict the behavior of the molecule in another way. Now, till now, you're interested that this molecule would be useful for cancer. But what your AI would do is, it not only helps you in predicting your cancer activity, it also helps you in predicting the other activities also. So that molecular behavior prediction is possible with your AI. Then finally, it doesn't require someone to give green signal to you. So system itself can take into consideration and it can take its own decision. So decision is one of the most crucial, you know, uh, characteristic of artificial intelligence. It can make its own decision once the feed is good, once the input is perfect. Now here I have given the definition here. Artificial intelligence are just your computer systems. They are designed to perform tasks just like your humans. As I said, I need a friend who is very much, knowledgeable related to my analytical part. That is nothing but your AI. What it is going to do, it is going to recognize the pattern. It is going to predict what would happen in future, what type of peak I'm going to get, at what retention time I'm going to get. It is going to predict, and it is a continuous learner. So I'm teaching it with 100 data sets. The moment I ask 101 question, it takes in two ways. It try to answer the 101, and it also take 101 as a teaching aid. So it is a continuous learning. And finally, the most important thing, what I already said, is it can make its own decision, so that automatically your load will be decreased. I hope I'm clear with the introductory part of your artificial intelligence and how it is related to the analytical part and how it is going to decrease the burden on the analyst. Hope I'm clear. So can I get some response from you? Are you following the things? Someone say yes. Let's try to move forward. Now, how AI techniques are used in pharmaceutical analysis. Before diving into the discussion, let me try to explain the ecosystem of artificial intelligence. I know many of you or most of you know the meaning of artificial intelligence, the machine learning and deep learning. But let me recap the things for my satisfaction, right? So artificial intelligence is comparatively a broader concept which makes machines to work smartly and intelligently, which means that they can think, they can decide, and they can solve the problems. Whereas machine learning is an integral part of artificial intelligence where we try to train the machines with giving some data and with giving some examples. When we try to teach them, it takes the inputs and it remembers those inputs and it try to replicate the same thing in the form of predictions. This is the meaning of machine learning. The third one is your deep learning. The meaning of deep learning is we generally don't teach the models here. We simply give the data. The data is so vast, the machine is going to read each and every data and learn the things on its own. Naturally, try to learn the things, and accordingly, it learns and try to recognize what you are expecting. You need to recognize and try to answer what you are expecting. So this would happen automatically. It just try to mimic your human brain. How you are thinking in the same way your deep learning models will be thinking. Let me give you a simple example. Now, the task is you try to develop a model to segregate the mails. What does it mean? To segregate mails means we are using so many platforms for emails, right? Let us assume that we are using Gmails. Now, daily I'm getting some 20 or 30 or 40 mails, out of which few mails are very important and few mails are spam, which are not at all related to me, which contains some sort of uh, uh, malicious links. Now, I want to develop a model to segregate before they are coming into my inbox. So this is my ultimate idea. So I can do the development in two ways. One is using machine learning and the second one is using deep learning. In machine learning, what you do is I will teach the machine like this is how your email looks like. If the same email is sent to multiple people in link you can get, right? You can declare it as a spam. This is the first instruction you are giving to the particular machine. The second instruction you are giving is if it is containing any links, malicious links, you can consider it as a fake or a spam mail. Then you will be using certain keywords like generally, many. If you are having such type of keywords without mentioning your name, if they are simply mentioning dear customer or dear sir or dear madam or in a related wording, you can predict that, okay, this could be your spam. So what you are trying to do is you are teaching the machine that the moment you come across a mail, try to read the mail. And if you find any of these features or characteristics, you can come to a conclusion that it is a spam mail. If not, it is a good mail. So what you are doing here, each and everything you are teaching it. So this is nothing but your machine learning. This is your machine learning. The second thing, what I'm speaking is deep learning. The task is the same. But here what I do is I simply give mails, lot of mails, like 100 mails, 1000 mails, 10,000 mails to the system. And what the system will be doing is it try to read each and every mail from first letter to the last letter. And upon reading each and every mail, after 10 readings, after 100 readings, after 1000 readings, it will be learning on its own, right? This is the official mail. This is the what do you call advertisement related mail, and this is spam mail. So accordingly, it learns by itself and accordingly can take decision. So it segregates. This is what happening in your Gmail, right? So inbox will be having different, folders like uh, you will be having social media, you will be having advertisement, you will be having inbox, you will be having automatically spams will be going into the spam mails. So this is the difference between machine learning and deep learning. Hope I'm clear with these terminology. AI is a broader concept. Machine learning is meant for uh, you know um, dealing with simple problems. Deep learning is the program which is used to address complex problems. Now these are the different models we have. As far as machine learning is concerned, we have decision tree, we have a random forest, and we have SVM, and we have KNN. So decision tree is something like we give particular characteristics, say like what is the temperature outside? Like, is it sunny or is it not sunny? The second one is, what about the sky? Is it clear or is it cloudy? So this is how you are giving the instructions. You can clearly teach the what do you call the system. Like, if the sky is cloudy, there may be chance of rain. If the sky is clear, there is no chance of rain. This is the first parameter. The second parameter is, if you can see sun, if it is sunny day, the probability of having rain is less. If you are unable to find sun because it is cloudy, the probability of having rain is less. This is the second parameter. Then the third parameter is, what is the humidity? If the humidity is high, probability of rain is more. If the humidity is less, the probability of rain is less. So what you're doing, you're teaching the system or the model in such a way that you give the parameter. If it is yes, you come up with this decision. If it is no, you come up with this decision. Now, the moment you give some question, right? What will it rain today? You're simply asking the question, will it rain today? Then it try to analyze what is the temperature out there. Is it cloudy? Is it humid? And accordingly, it will be predicting, it will be giving you the decision, right? Okay, as it is cloudy, as it is humid, as there is no sun, the probability of having rain today is more. So you can expect rain. So this is how the decision tree works. The same thing you can applicable for your drug also. So it takes into consideration few aspects, and you can predict that, okay, this drug could be toxic and this drug could not be toxic. This is how a simple decision tree machine learning model will work. The second one is your random forest. So what your random forest will do is, as I mentioned, you can imagine forest. So forest consists of n number of trees, which means that when the decision tree model, if you take 3 or 4 decision tree models together, it consists of a random forest. Like there, what you are doing, you are taking into consideration only one aspect or few aspects. Now here we will be taking lot many aspects. So say for example, if you want to declare whether this patient is a healthy individual or a diseased individual, what you take into consideration? His blood reports, his urine reports, and his normal symptoms, as well as the amount of biomarkers that are present there. So you'll be taking into consideration all these things, all the 4 concepts. Now, one, that is the blood sample, consists of one decision tree, urine analysis consists of second decision tree, and your normal symptoms consist of third decision tree, and the biomarker analysis will be consisting of fourth decision. So collectively, we call it as a random forest. Okay, so we are getting more information, so you can easily declare or you can easily predict this is a healthy individual because all his reports are within the specified limits. And he is a diseased person, his biomarker levels are fluctuating. His urine sample is having the borderlines. His blood sugar level is creeping over the limits. So such type of predictions we can come up with your random forest. Similarly, we have SVMs as well as KNNs also. This is what I want to make sure about your machine learning programs. Then we have a deep learning programs. As I said, it works similar to that of your brain. Like it will be having three verticals, like input, processing, and out. The input is what you require. And the processing is it try to analyze with the data or with the learnings what it already had. And accordingly, it will try to give the answer as output, just like your neural network, which are there in your brain. So we call it as ARN, artificial neural networks. We have plenty of models here like ANN, artificial neural network. Let me give you a simple example. Now, I'm giving instruction to the model saying that I have some 2 cents of land consisting of a double-storied building. This is first instruction I have given. First input I have given. The second input I have given is it is located in Chennai. The third input I have given is it was constructed some 5 years. So taking these inputs, what are the inputs? It is there in 2 cents. It is a double-storied building. It was constructed 5 years back, and it is present in Chennai, which is a metro city. Accordingly, try to predict the price of the house. So this is how you will be developing the deep learning models. Similarly, we can predict the solubility of the drug because we are already training it with n number of drugs with their characteristic features. The second one is your CNN, convolutional neural networks, where you feed it with the images. Like we train it with lot many, animal images in such a way that you will be having thousands and thousands of animal images in which you will be getting the model will be trained. The moment I give one image, say it is a cat image, it try to recognize that particular image and it will be giving you an input that it is a cat or it is a dog. Similar application can be done for your bioanalysis, like your cell line analysis. Like if I am suffering with cancer, automatically I need to go with the biopsy, right? Or I need to go with some what do you call CT scans or MRI scans, whatever it may be. So we train the model with these scans, these biopsy results. So when we train the model with all these images, it will be knowing at what stage the cell will be behaving or at what stage how the cell looks like. The moment I give the image of a patient who is suffering with a particular cancer, it try to analyze the structure of the cell inside the particular patient blood or patient's body. If it is good, morphologically fine, can declare, or the model can declare that the patient is absolutely fine, he is not having any cancer. But if there are any morphological changes in the structure of your cell, it can declare that patient morphology of the cell is deformed. There is a changes. It might be cancer. At what stage? That is possible with CNN. Similarly, we have other models like RNNs, LSTMs, autoencoders, GAN, as well as transformers. So go through this particular slide and you will be getting a easy idea about different types of models that are there as AI is concerned. Now, what I'm trying to say here is, basing upon your requirement, you can take or you can choose machine learning tools or deep learning tools. If your task is a simple one, you can go with machine learning. If your task is a complicated one or a complex one, you can go with deep learning models. No. From the starting of my presentation, I have been speaking about data sets. What is the data? Data is just like a feed or food to anyone. So without food or feed, we cannot survive. Similarly, the success of the AI model or machine learning model or deep learning model depends upon the type of data what we are supplying. If the data is good, if the data is huge and vast, we are going to develop a very good AI model or very good deep learning. If data is not that good, you are ending up with a false AI model or very cheap AI model. So data plays very important role. So where do you get data as far as analysis is concerned? The first one is the chromatograms are the main source of data for artificial intelligence. The chromatograms are being generated by your instruments like your UPLC, HPLCs, LCMS, GCMS, LCMS, etc. And apart from that, you have different spectra like mass spectra is there. You have NMR spectra, you have IR spectra, you have UV spectra. So all those things are being generated by the instruments and they are already present in your databases. You can get the information or you can get the data from the databases, or you can approach some reputed companies for the data. And accordingly, you can synthesize them. Along with that, you can get the data sets from metabolomic studies or proteomic studies or inomic studies. For example, I want to predict the pharmacokinetic studies. I would be taking the help of LC-MS data that is there under PK studies, who have already performed earlier. So some 1,000 people have performed pharmacokinetic studies that was there in the database. So I try to procure the databases and I use the data to develop the model. So this is how the data sources of AI comes from. This is where they are come from. Now I hope you are clear with the machine learning, the deep learning, the AI, as well as data. Now, how do you develop the model? How do you develop the model? That makes the sense here. Though we have plenty of complicated steps, I would like to simplify it with simple 5 steps. The first one is you have to acquire the data. As I already said, you can get the chromatograms, you can get the spectra, you can get the metabolomic data studies, you can get pharmacokinetic data sets from different sources. So this is the first and foremost thing. You need to get the clear, clean, and efficient and reliable data from reliable sources. This is first and foremost thing. The success of your model purely depends upon the acquisition of your data. The better data you provide it, the better model you are going to create. This is the first. The second step is data pre-processing. What is the meaning of data pre-processing? What is the meaning of data pre-processing? See, the data what we are getting might contain some dust in the sense not the real dust, unwanted things like the chromatograms may have unwanted peaks, unwanted interfering peaks, which have no role, which have actually nothing to do with. So what we try to do, we try to remove that. This is what we call it as noise removal. Similarly, the baseline should be sharp and it should be in a straight line. In some cases, you might be getting some sort of noise. So we have to remove that baseline noise or baseline correction has to be done. And then the peak what we are getting, it should be symmetrical peak. Sometimes you may end up with a trailing peak. Sometimes you may end up with a fronting peak. So peak alignment has to be done carefully. And with the help of the standards and with the help of your internal standards, we normalize the peaks. And accordingly, we filter the data. So all these steps are what we call it as pre-processing. Just we are removing the dust what is there in the product that is received. This is nothing but data first. You're cleaning the data. Unwanted things are removed. This is your second step. The third and most important step is feature extraction. So what is the meaning of feature extraction? Is though your chromatogram consists of 10 minutes runtime or 15 minutes runtime or 20 minutes runtime, you are interested with only few peaks, right? Peak of API at 10 retention time, peak of impurity 1 at 12 retention time, peak of impurity B at 17 retention time. Okay, those are only the features or the characteristics what you are interested with. You try to extract those important points only. That what is its retention time, what is its peak area, what is its what do you call a resolution. So all these things will be taken into consideration. This is what we call it as a feature extraction. Similarly, if it is a spectrum, what is its temperature ratio, what type of fragments you are getting, what is the fragmentation pattern. This is what we call it as extraction. Then accordingly, we will be designing the descriptors. What is the meaning of descriptors? The model cannot read the chemical structure. So we have to convert the chemical structure into easily understandable numerical value. So this is what we call it as a descriptors. So we are converting the image, the chemical image into understandable machine, understandable numericals. All these things are what we call it as a descriptors. And accordingly, we even try to find out the patterns, chemical patterns. All these things will be taken into consideration from the data what we have received. The third step is feature extraction. Now, the fourth step is really important. The features what we have extracted in the third step, we feed them into the model. We give to the model to learn. It could be random forest, it could be neural networks, it could be deep learning, whatever it may be. According to your task, according to your interest, you can develop a model, taking the extracted features from the data. So once it is done, you are ready with the model. The model is ready. You have learned, it has learned lot many things. Now you want to test it. So what you do is you try to test it with a unknown sample, unknown spectra, unknown chromatogram. And definitely the model is going to predict at what retention time you are going to get the peak. So according to my knowledge, the structure of this compound according to the supplied spectra is this. So that type of information will be generated by your model. That is what we call it as a prediction. If prediction is working good, then you can validate it. Validation, just like analytical method validation, we'll be comparing at different stages. We'll be trying to, slightly change the model and try to, see whether it is working properly or not. So this is how you can develop an analytical model. This is not a newer one. How you are developing an AI model, similarly we are developing here. But the data what we are supplying is the analytical related data. Okay. So the benefits of AI is in analysis is, as I mentioned, I would be taking some 15 minutes to half an hour to interpret one NMR spectra or one mass spectra. But the same mass spectra can be, interpreted within fraction of seconds by your AI. And the accuracy at morning 9 o'clock, I would be working much better because I'm fresh now. But at 11 o'clock or afternoon 2 o'clock, my brain will be draining. So the efficiency would be coming down. So that type of problem will not be coming with your AI because 24/7, 365 days, it will be working with the same sort of accuracy. You will have an enhanced accuracy. Moreover, you are taking lot many parameters into consideration rather than taking one or two parameters. The third one is all the complex tasks. I cannot do interpretation of UV, NMR, mass, IR at a time, but that is possible with your AI. At a time, you can interpret all the different spectra and try to predict the structure of the compound, which is generally not possible with humans. And finally, it is acceptable that we do commit certain mistakes. These are human errors. Human errors completely absent, or we can reduce it to maximum levels using AI. So these are the benefits of your artificial intelligence. Hope I am clear with what you call the development of AI models as far as analytical chemistry or pharma analysis is concerned. I hope it is clear. Yes, yes. Any doubts till this junction, or shall I move forward? I'll be taking 10 to 15 more minutes to complete the discussion. AI in chromatographic method development. So how we can use the AI in developing a new analytical method. So this is the traditional way. If someone is asking me to develop an analytical method for a particular compound, so what is the initial step I will be doing? I'll be going with the literature reviews. I'll collect each and everything about that particular, say, supposed to be. What is its chemical structure? What is its molecular formula? What is its pKa? What is its solubility? I'll take each and everything of it into consideration, and then I'll conduct few, preliminary tests. What is the solubility accordingly, and accordingly I'll be selecting the pH, and I will be deciding the buffer, and I will be modifying my mobile phase. Suiting the mobile phase, I'll be selecting my column. It could be C8 column, it could be C18 column, phenyl column, cyano column, or whatever it may be. It could be normal phase or it could be reverse phase. This is the third stage. Once it is done, I will be optimizing my mobile phase as well as stationary phase. I'll be running certain trials and I am coming up with an optimized trial. And accordingly, I'll be performing force degradation studies to optimize the resolution. And finally, the method is developed. Only the method is developed. Imagine for a single compound of sofosbuvir, for single API of sofosbuvir, I need to follow all these steps rigorously. So it will be taking some weeks. Imagine if it is for a single drug, for 4 drugs or for complex matrix, definitely it is going to take some weeks or sometimes months of time for developing a method. Because I'm just going with a trial and error basis. Though I have a theoretical knowledge, In reality, it may not work. In reality, it may not work. And in between, I may lose a better trial. So this is the problem with the traditional method or traditional development. But with AI, what it is going to do is, instead of taking literature review into consideration, it takes all the aspects into consideration. Like it knows each and everything about your drug that is supposed to be there. It has already data with it. And it already got some data related to the chromatograms. Some X people has used this mobile phase and this column. So they end up with the peak at so and so retention time. So such type of information was there with the AI already. So what it is going to do is, the moment you say that this is, I'm going to work on suppose Vivek, and this is its chemical structure, what it is trying to do is, it try to refer its library, it try to refer its data, and try to predict the conditions. Guy, you take this column and you use this mobile phase and you place it at this particular page. Definitely you are going to get the result. So that type of prediction is possible with your analytical development when you use AI. So it will be 60 to 70% faster. Instead of taking months time, within days you can complete it, if not within a day. So maximum 1 or 2 days or maximum 3 to 4 days, you can complete the method development even for a complex structures. So that is only possible with AI because you are already taught the model about the information. So it's not a science by sort of presentation. This was already reported by this team, Boswell et al. This was published in Journal of Chromatography A. What they have done is, they have taken machine learning rather than deep learning model. So they want to go with a simple task. So what they did is, they have collected the chromatograms, the structure of the compound, the pKa, the solubility, each and everything, and they trained the model. For example, see, if I take aspirin, the same aspirin structure, it is having the chemical structure, it is having the molecular formula, it is having the pKa. So it was fed with each and every information about your aspirin. Then what they are saying is, few people have performed method development for aspirin. Some have used C18 column. When they used C18 column and this mobile phase, they got retention time at this particular point. The second one, some other person have used C8 column. So with C8 column, with this particular mobile phase, they got retention time at this. In this way, they taught the machine. They programmed the machine. They developed the model with thousands of data sets, thousands of chromatograms. And after teaching the model, they have given a new chemical structure. And to their surprise, it has predicted almost 90% the retention time. For example, the retention time is 7 minutes. It was predicted as 7 minutes. But in reality, they have performed that. They end up with 6.8. So there is very close prediction. So that type of model was developed by Boswell et al. And others. It was published in Journal of Chromatography A. So again, I am saying the model success will be depending upon your data. So these people have, trained it with 1,000 of chromatograms. If you train with 10,000 of chromatogram, it will be working even better. So this is about your AI-driven chromatographic method. The same thing is even possible with AI integrated with QBD. Nowadays, we are speaking about quality by design, which is nothing but AQBD, comes under ICH Q14, which is a more of a logical way, more of a scientific approach to develop a robust method. But the problem with QBD is, compared to normal traditional methods, your QBD requires many trials. Though you are ending up with a successful method, you require many trials here because it is driven by statistical tools, design of experiments. Now we need to take into consideration what are the process parameters which are influencing, what are the critical quality attributes we need to focus on, and accordingly your design or software will be giving you the list of trials. Now with AI, no need of performing all those trials. It simply says that, okay, I already know critical process parameters and I already know critical quality parameters. attributes. Instead of going with 25 trials, you can just go with 5 trials. It is going to select the trials which are most important and which will give lot many informations. So that type of prediction is possible with AI integrated QBD techniques without compromising the quality of the analytical method. So that's about the method development integrated with AI. Then similarly, we have AI in spectral interpretation. So nowadays, interpreting the spectra is the biggest task what the people are facing with. Now, even it would be difficult for us to interpret IR spectra. Imagine interpreting NMR, MS, one-dimensional NMR, two-dimensional NMR, different types of two-dimensional NMR. It takes a lot of time and we require exercise to do. Imagine you just feed a spectrum into a system. If the structure is generated, how good it would be. So that is possible with AI integrated, spectral analysis. We have a few tools like CFM-ID, Sirius, and MetFag. These 3 tools are exclusively meant for mass interpretation. Even no need of giving the spectra to it. Just give the smileys to the input box. Taking smileys into consideration, it can predict the spectra also. And different modes, positive ion mode, what is going to happen, negative ion mode, what is going to happen. Such type of assumptions and predictions are possible with AI integrated mass spectrometric analysis. These are the tools like CFM-ID is there, Sirius is there, MetFrag is there. We can predict the structure, we can predict the pattern also, we can even identify the metabolites also. Okay, whenever you are finding free time, you can go through these tools, and they are quite interesting. I would suggest go with CFM-ID. Then similarly for interpreting your NMR, so automatically elucidates the structure without knowing the, the chemical shift values or correlations, the ppm levels, the shifts, all these things. Simply you give the information or you give the spectra, it is going to generate the structure of the compounds. Vice versa is also possible. You just give the structure and it is going to produce the NMR spectrum. You want one primary 1D NMR spectra, it is going to generate. You want 2D NMR spectra, it is going to generate. You want Quasi type, you want HSBC type, or you want HMBC type, it is going to generate all types of spectra as per your requirement. So few softwares are available like ACD Labs and Menova or Renova softwares are available for elucidation of NMRs. And our IR spectroscopy is mainly dealing with 2 regions. One is fingerprint region and the second one is your functional group classification. Now we train the model. Okay, if you get peak at this region, that could be an alcohol group. If you get this peak at this region, it could be a primary amine. It could be a secondary amine. It could be carbon-carbon single bond. So accordingly, we will be teaching the model. And the moment the model is completely trained, it is very easy to predict the structure of the compound from the the spectrum that is provided. So that is also available. So mass spectra, NMR spectra, as well as IR spectra, they can be used and they can be directly converted into easily understandable structures with a single click. That is possible with AI integration. Okay. So now whatever I have discussed are simple machine learning programs. Now why can't we integrate all the 3 together or all the 4 together? We have UV information, we have NMR information, we have MS information, as well as we have UV informations. Integrate all these models into a one system, that is a deep learning model. So it will be giving a beautiful output, like it would be 100% accurate. So that type of integration is also possible. We can predict the fragmentation, we can match the databases, and we can automatically detect what type of compound it can be. So that is all about your spectral recognition. And similarly, we can use AI in bioanalytical studies, like it could predict pharmacokinetic parameters by taking into consideration development of PK modeling, pharmacokinetic model. So when we develop a pharmacokinetic model, and once you give the information to it or instruction to it, simply you're giving the the structure of the compound. The moment you give the structure of the compound, it could predict this could be the Cmax, this could be the Tmax, this could be the area under curve, and this could be the clearance rate, and this could be the half-life. So thereby, anyways, we need to provide the real-time data with animal studies or human studies. But instead of taking 100 animals, you can end up with only 5 animals or few animals. Thereby, we can reduce the usage of animals as well as humans with the help of this predicting model. And even it is able to predict the metabolism. As I mentioned in proteomics or metabolomics, I know that the drug is curing your cancer, but what pathway it is taking into consideration, that is really very, very essential for filing the license or whatever it may be. So that type of prediction is also possible with the AI integration, and accordingly we'll be optimizing the dose. And even for bioanalysis also, AI integration will be helping up, because the problem with bioanalysis is there is a lot of overlapping with the matrix compounds or interferences or metabolites. So what this bioanalysis predicting model will be doing, it deconvolute. What is the meaning of deconvolute? Is when there is overlap, it try to segregate, and it try to give the peaks as 2 different peaks. It gives information about the first peak as well as the information about the second peak. So that type of prediction is also possible when we integrate LCMS bioanalysis with AI. And even we can discover biomarkers also, as in case of proteomics as well as metabolomics. Most importantly, with AI, we can predict the toxicity of the drug. So once we perform ADMET studies, we can perform the toxicity. Okay, this drug, though it is having a promising anti-cancer activity, it is equally toxic. It is creating some renal toxicity. It is creating some, rectal toxicity. That type of prediction is possible with AI once it is integrated. So few tools are there for such information. We can take the help of deep tox as well as PKCSM. And finally, can we integrate artificial intelligence in herbal and natural product analysis? Of course, we can integrate, because herbal medicine, when we speak about herbal medicines, the formulation consists not of a single plant. It consists of 4 or 5 plants. Imagine one plant is containing 90 constituents. How many constituents will be there for 5 different plants? More than 400, right? So separating 400 components or constituents at a single run is really tricky. So that is the main problem with herbal standardizations. So what we can do is, the AI tool can predict the biomarkers. So for this particular plant, for example, there is curcumin, there is ginger, there is black tea or whatever it may be. In each and every plant, there is one important biomarker. So that type of information will be predicted by the AI model. So accordingly, for each and every plant, we'll be taking 1, 2, or 3 biomarkers, and accordingly, we'll be designing a analytical method. So the moment you give the herbal formulation, if the herbal formulation is able to contain 3 biomarkers or 4 biomarkers, we can declare that the product is good. It doesn't contain any impurities. It doesn't contain any pesticides. So that type of standardization is possible. We can we can maintain the batch consistency with AI integration. So this slide is speaking the same. And finally, everything what we have discussed is in developmental stages. 100% reliable tools we right now don't have. So they are still working on it. As I mentioned earlier, the availability of data related to analytical work is very, very less. And even the data is available, they are there with the companies. So companies may not provide you the data. So that is the main challenge what we are facing right now. Technical challenges, we need large data sets to train a model, and the quality, you might end up with a chromatogram of very less quality. If you train the model with the same chromatograph, you might end up with a failure. Then how to interpret, how to develop the model as per the data sets. Technically, we have a challenge. And regulatory issues are also there. Whether the regulatory people accepts the method that was developed using AI, that is a bigger question. Still, A lot of talks are happening, and if they agree, then it would be very good. Regulatory challenges are also there, and more importantly, ethical concern. No company is ready to give their data because there is a privacy concerns, and moreover there is a bias also with the algorithm. So we have to convert the chromatogram, that images, into the numerical descriptors, and then we have to train it. And moreover, over dependence on automation is also a main problem. So these are all the challenges and limitations what we have as far as AI integration in pharmaceutical analysis is concerned. So what we can predict? So we can predict in future the HPLC that is running on its own. What is the meaning of running on its own? Now for operating an HPLC, an analyst is required, right? From switching on till to integration. So there are times where it is not possible for the analyst to be present 24 hours with the system. Suddenly there is a drop in the pressure. The instrument will be giving you a beep sound. Now the latest systems are designed in such a way that it will be giving beep sound for 2 or 3 seconds, then they automatically get shut down. But the problem with getting shut down is whenever we are switching it on, again we need to start from step A. So that is also taking some half an hour time or 40 minutes. Now, if you can develop a system that can predict the pressure fluctuations because of clogging, and it can stop the analysis, and it can wash on its own, and then it tries to start on its own, it will be very, very helpful to us. That is what the Thermo Fisher company is looking at, and they are developing the model. Thermo Fisher as well as Motors, they are working on it. They are working on an automated HPLC system that if there is a problem, automatically it stops, it troubleshoots itself, and again it restarts on its own without the intervention of the analyst or the technical person. The second one is the moment there is the there is a shortage of mobile phase, immediate sensors are alarmed, and alert is given to the pharmacist or the analyst so that they can refill the mobile phase. So you can control the HPLC system from the mobiles using certain apps from your home also. So this type of smart analytical equipment are going to come in near future. Maybe within 2 or 3 years, you are going to get smart HPLCs as well as smart LCMSs. And moreover, you are going to get a digital twins. What is the meaning of digital twins is you are working on a HPLC to get the results. What if you can get the same results without working on the instrument? This is the idea behind digital twins, which means that you will be having a software. What the software will be doing is it will be doing the same sort of analysis similar to that of the work that is already done by HPLC. So what we can do is we can avoid unnecessary trials. We can avoid the failure. So that type of digital twins in the analytical laboratories are also coming up in near future. And moreover, we can integrate industrial or industry 4.0 concept by integrating robotics, automation, as well as the AI into consideration. Everything will be automated so that we can decrease the human errors, thereby we can predict the perfect outcome, and then we can efficiently work with almost zero human errors. This is the future of pharma as far as AI is concerned. And vision is concerned, we can accelerate drug development when we integrate AI. We can enable real-time decision making when we integrate AI. And whatever results we are getting, they are 100% accurate if you are using AI. So this is a vision on which the companies are working now. And within few years, maybe within 1 or 2 years, we are going to get a very good softwares or very good models where we can perform analysis, or we can get the chromatograms, we can get the spectral analysis done within few seconds. So finally, I would like to conclude like the artificial intelligence is transforming the pharmaceutical analysis by integrating intelligent data interpretation, automating the method development processes. Advanced spectral analysis can be done. Taking into consideration all these things, we can predict bioanalytical modeling also. All these things are possible when we integrate AI in terms of big data into the automation as well as the industry 4.0 vision. So definitely the next generation would be much more interesting and much more fascinating, and we will be getting analytical results with a lot more confidence. This is how your AI analytical laboratory is going to look in future. Each and everything is smart instrument. Everything is working on its own. Autonomous laboratories will be there, and then we'll be having a lot of data sets that is being generated, and they will be integrated with real-time decision making. So these are the references what I have taken from. Thank you for your patient listening, and the floor is open for the discussion. If you have any doubts, you can ask.
Thank you, sir. Your session is very informative and knowledge gaining. I'm also one of the analysis faculty, Department of Pharmaceutical Analysis in Acharya Nagarjuna University. I learned more and more in this session. Especially what type of tools used in using AI in method development and validation using HPLC and spectroscopy. It is very good session for analysis people. Okay, thank you, sir. Thank you very much for accepting our invitation. And thank you very much, sir. So, dear participants, is there any questions? It's the question time. Is there any questions, queries, or doubts? Participants, please respond. Or you can post in the chat box.
Good morning, sir. I am Dr. Suvarna. I'm Associate Professor from Adichunchanagiri University, sir. So it's a very wonderful session, and nowadays we are having many more sessions on AI, but this is the rare one where we can find the AI-driven approach with respect to pharmaceutical analysis, and very wonderful and very enlightening session, sir.
Thank you, ma'am.
Thank you very much for this. And as you discussed, like some tools were there, like Sirius and CFMID. Sir, are these paid versions or any free tools are available with minimum
As far as
properties?
let me share my screen. You asked about CFMID, right, ma'am? So this is how the homepage of CFMID will be looking like, ma'am. The moment you give some smiles of that particular compound, and you give what type of mode you want and which edit type you want, it is going to generate the image, ma'am. Till this point, it is free. Apart from that, if you want anything else, it is subscription based, ma'am. I think this information is more than sufficient for initial interpretation or initial analysis.
Thank you, sir. Sir, any such AI-driven tools for interpretation of IR spectra?
IR spectra, they are working, ma'am. I have seen 2 or 3, but they are in what you call proprietary mode. And still they are in the what you call development. But they are giving some 98% accuracy levels. I'll try to convey to you, ma'am.
Sure,
The
sir.
names.
So still 2 more doubts are there. Please don't mind.
Yes, ma'am. Yeah, no problem.
Yeah.
Go ahead.
And with respect to AQBD, you have mentioned AI-driven approach. So usually what the tools we are using are design expert or a JMP softwares, something like that. And any AI-driven such tools are already available in the market?
So one example I've already mentioned, ma'am. People are developing the model. The problem, as I mentioned, for spectra, we have lot many information that is already available. For method development, we have a very limited amount of data that is available. Majority of the data is with either Waters Company or Shimadzu or Thermo Fisher. So they are trying to develop their own models and try to patent them. So in real world, there are no AI tools that are available for method development. But I have seen few papers, few people are coming up with their own models. Just they are saying that we have tried with these models and we are coming up with the prediction of 98% or 99%. Real-time tools are not available as far as I know.
Sir, as you mentioned about digital twins, what is your opinion about the digital twins for assay of formulations?
Yeah, that is an idea, ma'am. As I mentioned, without the real-time analysis, without using the instrument, what is there in my mind? You are trying to give the same information to the software. The software will try to analyze the parameters or characteristic features of the component, and as you mentioned, the formulation, and it is trying to predict. Okay, you may get the peak at this particular retention time. You may get peak at this particular RT or
Yes.
the peak area. That type of prediction is possible with digital twins. So accordingly, instead of losing our time in unnecessary trials, wasting the mobile phase, we try to do the same thing on the software. This is the main idea behind digital twins. The same concept as that of your, docking. So in
It is
docking
with
also,
respect to method development.
yes, it with respect to method development. Yes.
Thank you so much, sir.
Yes, ma'am. Thank you.
Dear participants, any questions? Or you can post in chat box also. Okay, so thank you, sir. Thank you very much, sir. Behalf of Acharya Nagarjuna University College of Pharmaceutical Sciences, I would like to express my deep sense of gratitude towards you, sir. We will keep in contact in future also, sir. Okay, you are the very eminent speaker in pharmaceutical analysis. Thank you very much, sir.
Thank you so much for that. Shall I leave, ma'am?
Yes.
Yes, ma'am.